Okay, so we already know that DNA directs the making of proteins. It is the master plan behind how a protein gets made because it contains the instructions. We're going to go more into detail. And you know that there's like an RNA copy made and it goes out into the nucleus, right? And then it goes into the ribosome, and then we get the protein being made out of amino acids. We're going to add a little bit more vocabulary. So new vocab word, synthesis, means making. So if you synthesize something, that's making. So this is all about protein synthesis. Protein synthesis is making protein. We're going to learn about 3 different types of RNA. And the first one is the one that we are already familiar with. It's the one that where you get the partial copy of the DNA that goes out into the cytoplasm and is read by the ribosome. That's called messenger RNA, which can be abbreviated with a little m, mRNA. There's 2 other types of RNA, but we have not learned about them yet. Okay, we will learn about them today or tomorrow. The process where we make an RNA copy of the DNA, we're going to learn the name for that. The name for that process is called transcription. Okay, transcription. Transcription is where you take the DNA and make the RNA copy of a small section. You make the RNA copy of a single gene. Because remember, what is a gene? A gene is the instructions for making. It is the instructions for making a specific protein. What happens after DNA makes the RNA? The RNA goes out of the nucleus. Where does it end up? In the cytoplasm. And what grabs onto it? Ribosome grabs onto it. And if this is the ribosome, the RNA starts feeding through, like it'll just go through like this as the ribosome reads it. Yes? And in the process, we make a protein. So we take the RNA and we use that code to make a protein. That process where we use the RNA to make a protein is called translation. Now, if you're all about abbreviating things, as I am, transcription can be abbreviated TXN. Okay? Translation can be abbreviated TLN. You do not have to use the abbreviations, but sometimes it's nice. Yeah? So translation is reading the code on the messenger RNA and assembling the amino acids into a chain. We already know polypeptide. That one is, what is a polypeptide? It's just the assembled chain of amino acids before it folds up. So it's like an unfolded protein. Protein is the amino acid chain after it has folded up. We already know protein. And so let's just take a look at the little picture here showing the process. And then we'll go into a little more detail on this process. So the DNA makes an RNA copy. This process here is called transcription. And that's the first thing that happens. The second thing that happens is that the mRNA goes out of the nucleus through the nuclear pore. So that's a movement of the mRNA into the cytoplasm through a nuclear pore. And then it goes into a ribosome, and the ribosome assembles the amino acids into a polypeptide, which then becomes a protein. And this process here taking place in the ribosome, all of that is called trans what? Translation. And it kind of makes sense because if you transcribe something, you are literally just copying it down. You know, back in the way back in the olden days, there were these monks that would sit in their monasteries and they would take a copy of the Bible or whatever other religious texts they had, and they would literally make copies of them because they had not invented the printing press yet. Yes? That would be called transcribing, where you literally just sit and make a copy. And if you have a strand of DNA, you are literally just making a copy of the bases using the complementary bases. Right? So you're transcribing, you're literally just making a copy. Yes?
mRNA and RNA are the same thing.
mRNA is one type of RNA. There are 3 types of RNA: mRNA, rRNA, and tRNA. We've only learned about mRNA right now. So the only RNA you know about is actually called mRNA. All the RNA we have talked about is messenger RNA. It's the one that takes the message of the genetic code out of the nucleus and into the cytoplasm. Okay, and this just again reiterates what we said above there, okay? In translation, a ribosome reads the mRNA code 3 letters at a time. Do you guys remember what we call those 3 letters on the mRNA? That's called a codon. So mRNA, messenger RNA, is the type of RNA that takes the genetic code for a protein out of the nucleus and into the cytoplasm. So nothing super new, just some new vocabulary around it, okay? All of these words with stars, you are going to want to add to your cheat sheet, okay? So if you want a tag for this page, you can tag that page. Again, there's some here, some there, some there, some there, okay? And then there's some exercises that we're not going to do right now. We will do them later today. For now, we're going to go look at RNA. DNA stores all of your genetic information. And actually, I'm going to introduce you to a new word here. All of your genetic information is called your genome. That is all of your DNA code. Every single base in your DNA is part of your genome. And back in 2000 and close to 2000, can't remember the date anymore, it was really exciting because they actually did what they call mapped the entire human genome. So, barring small little mutations, they actually knew the sequence of all the millions of letters in the human DNA code. And it took years and years and years to do that because you used to have to map it by hand. Back when I had a job doing some stuff with DNA, you literally had to chop up that DNA into little pieces, and then you would put them in a gel, and then you would apply an electrical current, and they would like move in this current, and they were radioactively labeled. Then you would put it on a piece of film, and the radioactivity would make the film like turn color. And you would literally have to go through and read the bases off one by one. You'd get a little code that looked like this, where all your A's were in the first column and all your T's were in the second column. You'd literally, you'd be like A, C, A, G, T, right? And you had to go through and read every single one like that. It was crazy. Yeah? And you had to do it all by hand. Now they have like genome sequencing machines that'll do it for you. But back in the day, it was all done by hand. So that is our genome. The next word you need to know is trait. A trait is a characteristic. So you need to know genome. You need to know trait. A trait is a characteristic. What are some of those characteristics? Well, there is height, blood type, eye color, hair color. Those are all traits. Yes. Always let me know if you need me to go up or if you need me to slow down or anything like that. Always let me know. Okay. Sometimes we're like, oh, what gets left out is how important RNA is. Without RNA, you could not get the message out to your cells. Getting real tired of playing the messenger here. Say hi to the ribosomes for me, says DNA. It's very cute. I don't know. There's actually like a video that goes along with this, but I've had trouble accessing the internet today. Like it just goes and it spins around and around. And so I'm providing my own voiceover. So it's not very good, but okay, you guys can live with it. All right, so now let us compare and contrast DNA and RNA. And if you are going to be taking biology, a compare and contrast question is something you'll often get asked to do. Compare means tell me the things that are the same. And contrast means tell me things that are different. And while we're talking about things that are the same and are different, I want to tell you about something that I saw a few people do on the quiz yesterday. You were to have given 3 differences between DNA and RNA, and a couple people did some stuff like this. They said DNA has U, and number 2, DNA has T. I got that backwards. DNA has T and RNA has U. Is that 2 differences or 1 difference?
1.
That's just 1 difference. So you can't say that's 1. You also could not just say RNA has U. You also need to compare with what DNA has. So you need to say both to show the difference. Okay? So what is the same? DNA and RNA are both found in living organisms, all of them. So bacteria have RNA and DNA, plants, animals, fungi, protozoas, one-celled animals. Okay? Now, there's something that is different. DNA is found only in the nucleus, whereas the RNA is found in the nucleus and in the cytoplasm. A lot of you told me that RNA was in the cytoplasm, but it's also in the nucleus, because where does it get made? It gets made in the nucleus. So it's actually both places. In prokaryotic cells, because they don't have a nucleus, they're just both in the cytoplasm because there is no nucleus. This is the same. Now, if you remember, DNA stands for deoxyribo, and then we have nucleic acid. And RNA is ribonucleic acid. What do they have in common? They are both nucleic acids. Nucleic acids are polymers. Polymer. So, for example, a nucleic acid what is the monomer for a nucleic acid? And I'm going to make this a little easier. I'm going to say nucleic acid is DNA and RNA. What is a single unit of DNA called? Nucleotide. Can you guys think of another single unit that gets put together in a chain? Yeah. Amino acids. An amino acid is a monomer. And what polymer does it make when it gets stuck together? Protein or polypeptide? So a nucleic acid is a type of biomolecule, which just means it's a molecule that's found in biological systems like you and me and plants and fish and everything, okay, that's just living things, okay? Proteins are biomolecules. Carbohydrates are biomolecules. Plants are, their structure is mostly carbohydrates. That's why we can eat them and get energy from them. So nucleic acid is the polymer and the monomer is the nucleotide. So are you responsible for the word nucleic acid? Yes, you are. So that's a star there. Are you responsible for biomolecule? Yes, it's just molecules found in living systems. They are large molecules found in living systems. So if you were to list all the biomolecules that you know so far, you know nucleic acids, which is your DNA and RNA. You know proteins. And technically, you kind of know carbohydrates, but not really. So we're not going to list that one. Carbohydrates. Yeah, let's put them in. Let's throw them in there anyway. Carbohydrates. And all carbohydrates, they are polymers. They are made of sugar. Sugars are the monomers. Although there's sugars that are more complicated too, but so we'll leave that alone for a moment. So the monomer for nucleic acids is a nucleotide. So both DNA and RNA are made of nucleotides. These guys are the polymers. And the nucleotide is the monomer. And the nucleotide has 3 parts. Tell your partner what those 3 parts of a nucleotide are.
Oh.
Okay, we got our phosphate, we got our sugar, we got our base. What shape do we usually give the phosphate? Phosphate is a circle. What shape do we usually give the sugar? Pentagon. And I don't know how I would do that with my hands. I'm like, I saw you doing the same thing. I was going to try myself, and I'm like, I don't know. Did you know that Ms. Bellier apparently has some sort of learning disability where she can't make her body follow instructions? You know, like if you've got like a right hand doing one thing and your left leg doing another thing. So at one point we were at grad and somebody wanted to teach her. They didn't want to teach her. They wanted to like do a little picture where both of them were sitting there like with little hearts, right? Like a little picture with a student and their teacher, right? You know, yeah, okay. She could not do this. She was like, I don't know. I don't even know what she did. I can't not do it because I've seen it, right? Like, and I know how it goes, but like she could not make her hands do it. And we're like, come on. We literally, Ms. Douglas, who used to be the dance teacher here, she's had to transfer to another school, but she actually like taught her because she is used to teaching people who maybe also have similar difficulties. So she's used to breaking things down. She did a really good job, and now Ms. Bellier can do it. So yeah. Interesting how different people's brains work so differently, because in math, she's genius. But like try and get her to follow instructions for like, you know, punch with this hand and kick with this hand. She can't do it. It confuses her. Okay, so here we go. There's our sugar, and then our base is kind of a rectangle. If you are interested, we do call them nitrogen bases, but the most official name is a nitrogenous base. Nitrogenous base. But all you really need to know is base, okay? But if you want to know the word nitrogenous, you can learn the word nitrogenous. Biology words are so long and complicated, aren't they? So much fun. Now, a lot of you told me on your quiz, you already know this, DNA is double-stranded and RNA is single-stranded. And you can notice that the two strands of DNA run anti-parallel to one another. You'll notice that the sugars point upwards on one side and the sugars point downward on the other side. See how we've got our little pointers there, there, there? That's anti-parallel because they're kind of running opposite directions to one another. The sugar in DNA is called deoxyribose. This is going to be enriched because if you are like, I don't want to learn that word, you can just call it the D sugar. Okay? D sugar. The sugar in RNA is called ribose. And we can just call that the R sugar, okay? So you can either call it D sugar or you can call it deoxyribose. You can call it R sugar or you can call it ribose. They are very similar. We'll take a look at what makes them different later, but for now, you can just know that they are different. Okay, and those two words are enriched. It's kind of helpful to know that for DNA, the sugar is deoxyribose because if you look at it, deoxyribo, nucleic acid. So it's the nucleic acid that contains deoxyribose. The sugar for RNA is ribose, and it is ribonucleic acid. So that's where those come from. Another thing you can notice is that both of the names of those sugars end in what? Ose. Can you think of another sugar that ends in ose? Glucose, sucrose, fructose, lactose. Those are all sugars. All sugars end in ose. That's just kind of interesting. You don't need to know that, but. So the type of sugar is different. The type of biomolecule is the same. Type of sugar is different. Type of biomolecule is the same. And a lot of people told me that the bases in DNA are A, C, G, and T, and that the bases in RNA are A, C, G, and U. And if apples go in the tree and the car goes in the garage, the car still goes in the garage with RNA, But you could say that the apple fell and it is now under the tree. So the apple is under. So A goes with U. A goes with T, A goes with U. C goes with G. What is the word that we say when we say C goes with G? We say those bases are complementary. Complementary, not complimentary. Why aren't you a lovely base? Your hair looks lovely. Thank you for smiling. I appreciate it. It's really only my first block that really gives me like, I'll throw out a joke and they'll all laugh. And I'm like, oh God, bless you all. My second block, they're like stony face. They're looking at me like, what is wrong with you? Why are you attempting humor? It's not funny. Oh, well. Yes, so sorry. It's okay. I think I'm very funny, and I'm the only one I really need to please, so it's okay. So there is only one type of DNA. There's only one type of DNA, but we've only learned about one type of RNA. There's actually 3 types of RNA. This is something that is different. We have mRNA. We have tRNA that kind of actually looks like a letter T. Yeah. And then we have rRNA. What does that look like? That looks like a ribosome, and that's because ribosomes are actually made of rRNA, ribosomal RNA, and protein. Ribosomes are made of ribosomal RNA and protein. And it is important, the capitalization. It is lowercase r and then big RNA. Yeah. Okay, so little r, big RNA. When you're doing tRNA, little t, big RNA. When you're doing messenger RNA, mRNA, little m, big RNA. You need to know the term rRNA. Ribosomal RNA is enriched. So what are the 3 types of RNA? There is mRNA, tRNA, and rRNA. So DNA stays in the nucleus, but mRNA has the ability to leave the nucleus. And actually, to be honest, the rRNA and tRNA also leave the nucleus. mRNA is so much smaller, it can fit through the nuclear pores. I think we already knew that, yes? And that gets it out to the cytoplasm where we have both amino acids and ribosomes. So it takes the message, it takes the genetic code out of the nucleus. That's our mRNA. And that's why it gets called mRNA, messenger RNA, because it is taking that message out of the nucleus. So not only do we need RNA to take the message out of the nucleus, but we also need rRNA to make the ribosomes to build the proteins, yes? Now, technically, the ribosome is made out of both rRNA and proteins, but that's just going to confuse you, so we're just going to leave it at that. Okay. rRNA is ribosomal RNA. Ribosomes are mainly made from rRNA. So ribosomal RNA, that is an enriched word. rRNA is a regular word. Could you guys close the door for me back there, please? Maybe Cody, you could close the door for us. Thank you. Where are ribosomes made? This was a tricky one for a lot of you. They are made in the nucleolus. So in the nucleolus, ribosomes are made here. Now, what would our cell look like? Our cell would be like this. This would be our cell. In the cell is a nucleus. Inside the nucleus is the nucleolus. So inside the cell is the nucleus. Inside the nucleus is the nucleolus. And in the nucleolus, that's where ribosomes are made. Now, what about tRNA? We got our last type of RNA. Transfer RNA. What is it transferring? Well, we don't know yet. We're going to find out in a minute. Transfer RNA, this is an enriched word. But tRNA is a regular word. tRNA, its job is to grab amino acids grabs an amino acid and it brings the amino acid. Where does it bring it? It brings it to the ribosome. So we don't just hope that the amino acids end up at the ribosome to get assembled into a protein, the tRNA brings them there. And you know what's lovely? Watch this. Do you see how on the mRNA, which is in the ribosome, there is a codon. What does a codon code for? 3 letters tells you 1 amino acid. And how do we get the correct amino acid? Well, the tRNA has a code that is complementary to the codon, and it is called the anticodon. The anticodon is complementary to the codon. It's a 3-letter code. And if the anticodon is UAC, then perhaps we get the amino acid serine. That means that the codon AUG tells us serine. So any tRNA that has UAC will grab a serine out of the cytoplasm. So there's just amino acids floating around everywhere. And if I was a little anticodon and my feet said UAC, I would say serine. And I would grab a serine and I would take it to the ribosome. And then my feet, which say UAC, would match up with the codon that says AUG. Yes? And that's how we get the correct amino acid in the correct place at the correct time. Okay? So you know that on the mRNA, we have the codon. So the codon is on the mRNA. The anticodon is on the tRNA. And you know what's nice? Anticodon has a T in it. So if you get confused, if you're like, is the anticodon on the mRNA or the tRNA? You'd be like, anticodon, it's got a T, it's on the tRNA. And here's a little question. What is the complementary DNA code from which this mRNA code here was made? What was the complementary DNA code? We've got our RNA code AUGCCA. What was the complementary DNA code? So we got AUG. So what would go with that in the DNA? A goes with T, because this is DNA now, right? And then U goes with what does U go with? A, because apples go under the tree as well, yes? And then we have a G, which goes with C. And then we have a C. What does C go with? G. And then another C, which goes with G. And then this A T. So that's our DNA. I'm going to stop. I'm going to get rid of these circles because that's just making things messy. What will be the next tRNA anticodon? We've got one anticodon here. We've got the UAC anticodon, and it's going to match up with this AUG codon. What's going to be the next anticodon on the tRNA? What matches C? G. What matches C? Well, that's another G. And what matches A? Because it is tRNA, that's going to be U, and that is our anticodon. So this is the codon on the mRNA, and the GGU is the anticodon on the tRNA. And then there's actually a chart where you can go and look up what codon gives you what amino acids, right? You don't have to memorize that kind of thing. You just go and you look it up. So the DNA has a triplet. The mRNA has a codon. The tRNA has an anticodon, and that anticodon also gives you an amino acid. And that's how you get from the DNA code all the way to the protein. Crazy, yeah? But it works so well because everything matches up perfectly. When all the amino acids are joined together, they form polypeptide chains. Those are our polymers, and they eventually fold up to make proteins, which are biomolecules, and they have many jobs. Can you guess the jobs for each of these proteins here? What do you think is happening in number 1? That is, there's our Amazon little arrow thing. That is a transport protein, and it might get something from outside the cell to inside the cell, or from inside the cell to outside the cell. What do you think, what type of protein do you think this one is? Structural. Yeah, we got a little construction hat on, the hard hat. Structural protein. I'm not quite sure what they want to happen here. I'm guessing this is an enzyme, but I don't know. I don't think that's a very good drawing, like to give you an idea, but maybe. What do you think is happening in this one? That is protecting. What do we call those? Antibodies. And I think that's it. There's a quick amoeba quiz here. If I have 8 DNA nucleotides. This is tricky. And the reason it's called an amoeba quiz is because the video that goes along with this, if you want to watch the video, the QR code is at the beginning of the thing, is called the Amoeba Sisters. Have you ever seen anything by the Amoeba Sisters? I love the Amoeba Sisters. I'm so sorry that you couldn't watch them today instead of listening to me. But like I said, I've had trouble with my internet all day. If I have 8 DNA nucleotides, how many DNA bases do I have? If I have 8 nucleotides, how many bases do I have? Each nucleotide has 1 base, so I have 8 bases. How many base pairs do we have, like matching? You know, like an A and a T or a C and G. How many pairs? 4. Yes? And you can see here, if we had just this here. Oh, and you guys can't see that one. Oh, yeah, you can. Okay. If you had just these 4, that would be 1, 2, 3, 4, 5, 6, 7, 8. There's 1, 2, 3, 4 pairs. And you guys are going to fill in the complementary DNA bases for this strand. And then you're going to fill in the complementary RNA bases. Uh-oh, I did something wrong, didn't I? Where did I go wrong? Ah, that should be a U. 1 mark off. Man. Okay, so you guys are going to add everything from this to your cheat sheet. So that's going to be pages 56 to 64. And what was the other one that we just had? 53. Now, to help you do your cheat sheet, would it be a good idea to do the exercises? Probably. Are some of you going to go home and not do the exercises because you don't know how to do homework for more than 5 minutes? Yeah, that's going to happen for some people. Because you don't know how to do homework for more than 5 minutes yet. That's not going to get you very far in grade 10 and 11. And you guys are at the end of the first semester of grade 9. So I'm going to put it on you. I'm going to say you should, for homework, have pages, oh, that's a lot, 64 to 66. And 54. just to up your game a little bit. You should have a little bit of stress this last week. Not bad stress. There is bad stress and there is good stress. Good stress pushes you a little bit. It gets you going. Now, there is a reason I don't give projects, because every other teacher gives you projects, and I figure I can be the one teacher that doesn't. So you guys are not lacking in projects, I take it. You probably have enough projects? Yeah, that's what I figured. So I can be the teacher who's like, nope, you get a test instead. There we go. But the nice thing is, you know, it's all over on Friday. Yeah? How are you going to get your test back? You have two choices. You can come in on Monday or Tuesday to get your test. Okay, I have a doctor's appointment on Wednesday, so I will not be here on Wednesday. I booked it so that, you know, what? What? Oh, okay. Yeah, I just booked it because I can like take the day without having to prep for a sub, right? So you can come in on Monday or Tuesday, or you can check your result on My Ed, right? Which doesn't let you see the whole test, but it does let you see your mark. Now, if you don't know how to get onto My Ed and check your mark, you can go on right now and try and check your mark, and I can help you out if you need a little hand. Okay, you might want to figure out if your My Ed account is working, because we've had some issues with that for a couple of people where it wasn't working. All right, so make sure you've got that. But like I said, I will be here Monday. I will be here Tuesday. I start at 10 o'clock, okay, because I don't work first block. Okay, I work part-time, sort of part-time. But that doesn't mean I didn't spend 9 hours making grade 12 tests on Sunday. So, yeah. Part-time, fun. Part-time, where you work every weekend. It's okay. I get my big holidays, so that's all right. Yeah. Just too much going on.
Busy, I have to, or I won't be able to do things
Right.
which I need to do to actually get on work.
Yes, I understand.
Yeah, no, I do not. No, I take picture of me, please. I've limited storage. I just want to yeah.
Oh, yeah. I figured
You want a picture, man?
I finished saying this podcast.
Got back on it.
I'm almost ready to be done.
No, not you. I took a picture.
What are you doing?
I see my camera now.
That would actually be a good thing to do. That's very good.
Good. I love you, like, when how you guys met outside? There was like that I don't know what to do. Loud. I don't know. I don't know on the outside I have all the guns, but this is so good. I think I'll get ready to get
Not like this.
Weak. I want to write my stuff. I want to draw, but I can't.
Oh.
Oh my god. Okay.
Five seconds. Tell me answers. 3, 2, 1.
I'm sorry, I'm trying. I sketched out two and gave up. But still, I can sketch out two. I can't even. No one else.
Five pictures. No.